them down into different clusters. We started breaking them down into functional feeding groups, into defense status, in the sense that if you have a shell, you would be defended from a predator. If you didn't, can we start looking at that as a way in which the viruses move? And we didn't see a clear signal, but we're in the infancy of our ecology. And when I speak to ecologists, they were like, haha, your data sets are very small, not enough stats, which is not surprising. We're virologists. We can't generate those large data sets that quickly. So we are getting there. We are using some of these concepts in ecology already, and we're building on this, and we're getting there. The other things that we start finding is we all know, and there is a big hypothesis, that we actually all came from an RNA world. RNA first, then DNA, so forth. We're finding relics of viruses, which are chimeras, in the sense that they are all DNA genomes, but they encode proteins that we only find in RNA viruses. And we started finding these in boiling hot springs. We've been finding them in sand. We've been finding them in the Antarctic as well, and we'll talk about that shortly. So when we start looking at the diversity of things that we found so far, these are things that we know exist on the planet. We've really well characterized. All these branches here are things that we've discovered in the last 5 years. And if I was to put that into a scale, probably would have to generate another 1,000 families to house those individuals. And that's how much diversity we've just uncovered in 5 years. Okay? All right. Now let's move to the main area. So earlier I said those parrots that had lost their feathers was the reason why I ended up in the ice. So David Ainley, he's a penguin. I call him a penguin because he's as hardy as a penguin. He's lived with these animals for at least 20 years, 20 seasons. He knows them really well. What is interesting is one season he noticed that the chicks did not have any feathers, and it worried him quite a lot. And this is at Cape Crozier on Ross Island, one of the windiest places next to Cape Adare is probably the windiest. But on Ross Island, the windiest place is Cape Crozier. There we've got 200,000 pairs of breeding Adelie penguins, and this one season he noticed this really, really weird pattern. So he came out to see me and said, hey, you work with animals that lose feathers. Can you try and figure out what's happening with these poor birds? And before I knew it, I was out in the field with their team trying to sample things. And the Antarctic is magical in its own way, especially McMurdo. You get there, the Prairie Lab, you end up meeting people, and before you know it, you're sampling all sorts of random things which you didn't even want to, but it just happens. Sampling ponds, you meet a geologist, and you're like, oh, let's look at layers that existed 5,000 to 8,000 years ago. And then, oh, you see emperors like not far from where you are. You're like, okay, let's do emperors as well. And then skuas are feeding on everything, so they're in your face all the time. And these cute little Waddell seals, they're just so cute. They're just like, come on, sample me now. You're doing everyone like, come on, me, I'm the next one. So, before you know it, you get into that realm and you get sucked in and it's too late to move out. So with Joe Levy, who's based now at Texas, is now based in Texas, he would give a seminar where he was showing something really bizarre. And he said that he's got all these layers in the dry valley, and there are these layers of algal mats, so mainly cyanobacteria. And with climate change, it's receding really fast, but there are rivers running in the summer next to it. And it seems like the algal mats are seeding the rivers with whatever was trapped in the last 8,000 years ago. And I was like, wow, that's interesting. Because if I think about evolution of viruses, 8,000 years of viral evolution is a lot. Like, we should be able to see a very clear signal. So I asked him, can we sample these layers and then we sample the water? If we see completely different communities, we might be able to try and track something out of this. And then I also got worried that if we are leaching things into a well-established ecosystem, what are the dynamics of this new entity we're dropping in? Could be cyanobacteria, could be viruses, could be anything else. So we started looking at some of these things, figure out pretty much cyanobacteria in these layers. He dated some of these layers to about 7,000 years old. We find a heap of viruses in there. They're still intact. I think it's pretty much the ice has done a wonderful job for us to keep them intact. We do not see any deamination in them, which is great. You know, as soon as you start talking to any ancient DNA person, they're like, hey, time, deamination, double strand breaks, you know, that's the clear thing. But it looks like these things are really, really well conserved. We can identify a whole lot of viruses. We also identify, remember I talked about those little viruses that had 6 segments and then 8? Because we worked with those plant viruses, we were able to identify very similar molecules in these layers as well. So those viruses that we're seeing in nature infecting plants were present, possibly similar, or relics of them, about 7,000 years ago. So it's not something that's happened overnight or recently. They all have a common region, and that's what we can use to recognize. They all encode one protein. The other protein needs to bind to this very, very common region to enable it to replicate. So this has got us thinking, and this is where we're trying to move forward a little bit, is, are the melting permafrost seeding the ecosystems, and what are the consequences of this seeding effect, especially if you have something trapped? And we all know about what has happened. You know, someone found smallpox in their freezer, and it was this big outcry, and we're talking about 100 years there, just short of 100 years. You go to Siberia, mummies are being exposed, and I can guarantee you, if you take a tooth out of that mummy, drill into the root canal and analyze it, if it's dated at about 1800, 1800, when it died, it's highly likely it died of smallpox. Okay? And so what is happening with some of these organisms that are being uncovered due to climate change? Are those pathogens that are trapped going to be detrimental to society and the ecosystem today? So those are some of the questions that we're kind of trying to get to. I work with a very used to work with, he retired, a very brilliant English gentleman who was based in New Zealand, and he was an expert in diatoms, but he also had a passion for cyanobacteria, Paul Brodie. And he'd done a lot of sampling in the Antarctic. He was in the same department as I was, and he's like, come on, let's have a look at something. So he kind of sent me a whole lot of samples he had, and some of them were from these ponds on the McMurdo ice shelf that were like really alkaline. And in there we found lots and lots of cyanobacteria, lots of diatoms, and when we analyzed them for viruses, they're full of really interesting viruses that we've never seen before. Again, we're in that realm that we actually have no idea what's going on with these viruses. And then we go to Dave Ainley's samples. These penguins, they look cute, but they're not. They're cute to look at, but they hurt because they love to flip a basho. And I am quite dark, and I come back with bruises on my leg. And so these animals are cute at a distance. But they are full of viruses. We actually don't even know what these viruses are doing. There's so many of them. And we've got about 60 viruses we found from the fecal matter that we have no idea. And so we're busy trying to figure out what these viruses are doing, how prevalent are they in these animals, are they at all causing any disease? And these are the unknowns. What is actually happening with these animals? So with these unknown viruses, if I were to put them in these big phylogenetic analysis with DNA, single-stranded DNA viruses, this group's here, and these groups here, and that there, we know everything about. All of these, we have no idea. Everything that is in color in those areas is everything that we found in the Antarctic. So the diversity we find in the Antarctic is almost equal to the diversity we're finding elsewhere, indirectly. So it's not that the Antarctic is a unique continent and things there are going to be completely unique. And that was what my idea was when I went there. I was like, we will find some very unique signatures there. No, they're not that unique. So within the Antarctic, we decided to look at other things. And this was something that I was passionate about. And this is papillomaviruses. I kind of, for my PhD, was heavily involved in designing vaccines for papillomaviruses. So it was this kind of going back 15 years to where I started. In the Adelie penguins, we identified a papillomavirus. Papillomaviruses have never been identified in penguins. The only bird papillomaviruses we know about are in crows. We know about parrots and finches. That's it. We know nothing else. We know about 2 of them in turtles. But if you look at the analysis, phylogenetic analysis, this is what we find in a lot of the mammals. Some of the human ones are scattered in there. If you look at that, that entire clade is basal to what we find in mammalian systems. We've just found a second one from the last season, another papillomavirus, which is also very divergent. And so we're now figuring out that actually these animals in the Antarctic also have papillomaviruses, which is kind of interesting. The Waddell seals that we've been working with Jennifer Burns, we've been taking vaginal swabs and nasal swabs. In the vaginal swabs of these animals, we get about 40 swabs back from Jennifer Burns every season. So far, in 2 seasons, we've identified close to 10 totally unique papillomaviruses that don't at all cross each other at all. So in these animals, there's so much diversity of papillomaviruses as well. So this is kind of areas we're dabbling in with things that we know. The other thing that we found is polyomaviruses. These are viruses we found in a small group of birds: geese, butchbird, crow, canary, budgerigars, finches. We found them in Adelie penguins. We've also just now found them in Waddell seals. Up here, there is one found in California sea lions. It's very similar to the one found in California sea lions, variation of about 15%, which is kind of what we estimate, because these viruses have co-evolved with the hosts and co-speciated. So it's not surprising that we're seeing some of these signatures. But then one thing threw us out, and that is Antarctic fish. We've been working with Bill Davison, who's based at University of Canterbury, where I was, and he's interested in fish physiology. And at some point I got talking to him and I said, hey, why don't you just give us some of the offcuts of your fish, things that you're going to throw away once you've done your fish physiology study. And he brought me a whole lot of fish. We looked at this, the Nortons, and it was kind of one of these weird things that happened. I just mentioned to you polyomaviruses that we found in Adelie penguin and the Waddell seal. Polyomaviruses have never been found in fish, ever. People thought they don't even exist in fish. But then three research groups independently at the same time found polyomaviruses in fish. Chris Buck at NIH decided he was going to go to a supermarket and buy some sea bass and find out what's in his sea bass. Jerry went to an aquarium and he found this virus in a guitar fish. We found it pretty much in this Norton in the Antarctic, which was fished out. It lives at about, I think, 300 meters or something like that. And that group, here are all the avian polyomaviruses. Look at all these fish polyomaviruses, and we're actually looking at a gene that enables it to replicate. So we're not even looking at the gene that enables it to actually internalize into a cell or bind to a cell. And around the same time, we started doing a little bit of analysis, and someone had put in a whole lot of transcriptome data of a spider from South Africa. And we noticed that actually some things in the arthropods stick out like a sore thumb. And in New Zealand, we've just finished looking at, not started, sandflies. And we find an entire new clade of polyomaviruses that sits here. And completely discovered fortuitously. We have no idea what it does, whether it's transmitted to humans or not. We're finding these things. But when you look at the capsid protein, which these viruses need to attach to the cell surfaces, get internalized, the fish ones stick out like a sore thumb. And you can see some of the avian ones kind of group with things that we find in mammals. But this is kind of one of the things that we find really, really striking in these kind of things. And we're trying to develop models of evolution where these viruses have evolved, recombined with certain things, and then what we're seeing today. So we're kind of working also in some concepts of ancient evolution. And with this study, we presented our hypothesis on what has happened with polyomaviruses evolutionarily early this year in one of in PLOS pathogens. Now, this is something that is really weird. There's a virus called Aniela Virus and a group of viruses called Aniela viruses. People find them in large quantities all over. They infect a lot of mammals. At least we think they infect them because we're shedding them in large quantities. Very simple genome architecture, 3 genes. These guys here are either found in California sea lions or harbor seals. All of these have been found in the Antarctic in Weddell seals. So you can see in one season in the Antarctic, we've just completely expanded this knowledge base of what's existing there. And we're now trying to figure out why is there such a high diversity of these things? What are they doing? Why are they being shared at such a level? One thing that we notice that there is a lot of slippage that takes place there because there are lots of GC, AC rich domains. So during replication, there is slippage taking place, and I think these guys are evolving very rapidly through this mechanism of slippage and exploring sequence space. Circoviruses, going back to feather loss in Adelie penguins. So we've been kind of monitoring these penguins for a while. We think, can we find a circovirus? Is there one? Is there not? And lo and behold, we recently found finally a circovirus which we think might be responsible for that feather loss. That circovirus in Adelie penguins is very, very neatly clusters with all the circoviruses we find in birds. We've also found one in Weddell seal, which is really interesting because it's got long branches, which is not surprising. But these viruses here have been found in freshwater fish. So we think there is a new clade here that's coming out, which would be probably an aquatic animal type that pretty much lives a lot of its life in that environment. So we're trying to figure out what actually is going on in that area. So eventually, this is where we want to get to. We've been finding Aniela viruses in Weddell seals. These guys feed on the placenta of Weddell seals. We find the same virus in these two every now and then. We find viruses in those guys. We find the same viruses in there because they feed on Adelie penguins, especially the chicks and the eggs. Now, can we start using viruses as proxies of trophic interaction in systems where we can't see those interactions? And this is kind of an area we're beginning to dabble in. The fact that we're finding these viruses that connect these guys, viruses that connect these guys, even though they live in the same ecosystem, we do not find the virus in here. So that's telling us that there is a very direct link in that trophic interaction. We do find viruses that are in there in all of these three. So we've been looking at the fish gut is a very, very good way for us to sample what is going on in there. Instead of taking water sample, we can just take the fish gut, and that should have large quantities of pretty much everything concentrated. What we see in those permafrost layers, 7,000 years old, and that is here in Victoria land, this is the dry valleys, we do not see at all in any of our samples. So we see a very clear transition on things that have been trapped so far. Things that we see in this McMurdo ice shelf, we do see in some of the fish samples in the guts every now and then. So we're beginning to start deciphering this at an ecosystem level. We've done it from a very, I would say, kindergarten fashion at the moment. We're still in preschool, but we are migrating and we are learning. Hopefully we will graduate to a proper school, and the ecologist will accept some of our concepts and work with us and teach us. And together we actually hope to get further up and actually start answering some very fundamental questions in our system. So when we look at the viral landscape that we started with, this is where we are. It's a very noisy background with a little bit of signal in there. We're trying to improve and get to this level here, where we can see the signal in that noise. But there is a problem. Viruses evolve really fast. I would like us to get to this level here, but I don't think we will. Because by the time we get there, those viruses would have evolved and put us back at that point. And so this is the game we play with viruses indirectly. You know, you could have a virus in existence today, 30 years from now, it's extinct. It's been actually out-competed in a population, it's gone. We are studying viruses, they're evolving at such a high rate, it's phenomenal. So we're hoping we can get to at least that, and if that's a resolution we can get to in our landscape, we would be content and be able to actually predict spillovers in ecosystems, predict certain fundamental changes, or what viruses can do to mitigate or offset an entire ecosystem. Okay? So a lot of this work spans many groups, many areas. A lot of the work I can't do on my own. So we've got collaborators that come into play. We have a very open lab system. Science is free, I believe, and anyone is welcome to play. So a lot of students have been involved in these projects we've been doing and we carry on doing. There are people in my lab who have been involved. I have a cohort of a core group in South Africa that I work with. I have an adjunct position at the University of Cape Town. So we have a team there that we interact with very closely. We've got an American team here, obviously with Maya and Karina being pretty instrumental here, but a whole lot of other people that we've been working with very closely. Within the Australasia setting, we've got a cohort working in Australia, etc., working on a lot on plant viruses, but we've also been looking at geological samples. We've been looking at volcanoes, and inevitably, before you know it, if you're crazy like me, people would say, hey, how about we look at that? And you're like, oh, okay, yeah, it'll be fun. And then, you know, you start working with geographers who want to go out and bring you boiling mud and all sorts of things. So it's actually fun to work with communities all around. And this group here has been also really cool. These two are veterinarians in Poland, and they've been kind of working with us really closely. And Claudia has now moved to USDA here, and she's basically opened up a completely new avenue for us for testing for avian influenza. So they want all the swabs we bring back from Waddell seals and all animals and even migratory birds. They want to test them for us, which is really cool because the whole group is expanding, the knowledge is expanding, the science is being better understood, and that's where we want to be. Cool. And this is kind of something that's really important about single-stranded DNA viruses. Thank you very much. Yes?
I'm very impressed. I wanted to ask you about, well, first of all, I see you have Terry, one of our alumni, at the bottom of your slide there. And the other question I wanted to ask you was, have you looked at lunar samples and looked for viruses in there? Have there been contained enough that you could actually analyze?
Sorry, in?
Lunar samples?
So we haven't looked at lunar samples. So there are a couple of things with the lunar samples. There are few people who are really interested in there. Now, the only thing is, how have they been packaged and stored? And this is the same thing that we have in the Antarctic. So that layer that I mentioned that we sampled, which is about 7,000, 8,000 years old, I cannot, I'll use this phrase, maybe you can edit it off that thing, I cannot put my cock on the block for it. Because we get katabatic winds coming in, so you could have viral particles that are in that circulation. I can say that I've tried my best. Even with the vessels I'm sampling with, I might have carried them from elsewhere. So that's the same thing we have with lunar samples. Now, people have been wanting to look at meteorites, but then you've got an impact, and we do know that energy at impact will not let any nucleic acid survive. People have done those tests. But then a lot of people want to look at colonization of those meteorites. So that's an area that we're about to start dabbling in. Once a rock has landed, how quickly does it get colonized, and who colonizes it? And can